Yeah, good evening, everyone. Okay. So, okay, quick question. This is ORS solution, ORS, right? The solution that someone has a stomach upset or something like that, typically this is given. This is ORS solution, and how does it work? Oral rehydration solution comes in this sachet form. As a powder, you can mix it in water. Okay. Notice this is a life-saving, low-cost medical treatment. Can be technically prepared at home, purchased as a packet, or sometimes this is also comes in a juice form. It's like energy drink. Yeah, this is actually very different from an energy drink. In fact, this is one of the treatments that has literally saved millions of lives. Okay, and the objective, yeah, this is usually salt and sugar. So why salt and sugar? And we want to understand that. Okay, that is the objective of today's discussion, and in the process we'll get a very good idea on all the intracellular, extracellular fluid, what is going on in a cell and things like that. Okay, so this is one question. Fine, this is salt and sugar solution, special mixture of water, sugar, that is glucose, and some electrolytes, like you have sodium, potassium, and things like that. Probably sodium chloride, potassium chloride, salt. Okay, this is used to. So why does this work? Okay. At this point, let me also show you some history. Okay, so I think the last class, not the last class, that is the last time we did a discussion like this. This was on lychee fruit poisoning. Okay, where the problem is hypoglycemia, low sugar, low blood sugar. The solution is the moment you give blood sugar, automatically the children recover. Right? That is a story. Here, it's a very different problem. Okay, there's a problem with dehydration, and we want to understand what this dehydration is, right? What is going on in that? What do we mean by replenishing them and things like that in finer detail? So in one line, sugar water worked in lychee poisoning. Sugar water will not work for things like diarrhea, specifically that due to cholera. And okay, now glucose enhances the absorption of sodium water in the small intestine, even when experiencing diarrhea. Okay, I suppose, are you referring to this mechanism, Isha? Yeah, that is, so glucose enhances. So no, this is a very interesting case. Okay, in fact, this is a very, very simple summary. This is kind of given. The answer partly is given in there, but this is like a trailer. Right, glucose enhances the absorption of sodium water in the small intestine. It so happens when people have diarrhea, specifically due to cholera, they are the body is running out of water and sodium. This is they are getting lost in the diarrhea, right? So sodium is lost, water is lost. If you give them water, it will not enter the body. That is, it will be in the GI tract, it will be in the gut, it will not enter the body. If you give them sodium and water, again it will not enter the body. Only if you give them sodium and sugar solution, salt and sugar together in water, that enters the gut, that enters the gut layers, that can enter the body. Otherwise it cannot enter. This is due to the fact that sodium, there is something called co-transport. This is quick, very, very quick analogy at this stage. There is something sometimes it's a very common thing in colleges and all that. People would like to go to watch a movie, but if any one person kind of backs off, then the movie plan cancels, right? So in this case, the glucose and sodium are the two movie goers, the pairs here. Okay, so if both of them are there, they'll go to the movie, but only one of them are there, the movie plan is terribly spoiled. Very briefly, that is the trailer story of this. And let me also motivate this from a very different perspective. So ORS, this is a solution that has literally saved millions. And in fact, by the end of this, we roughly want to understand this to some extent. Okay, if you have to design, how would you make it? Like, so roughly glucose should be the glucose and sodium similar molar levels, similar numbers, number of molecules is similar, molecules per liter. Potassium is lower, and then you have chlorine, and then why do you also give citrate, right? So we want to actually understand this table reasonably well, but beyond that, notice history definition. 1980s, the oral rehydration therapy meant only what is prescribed by World Health Organization. Okay, but notice 1953, Hemendranath Chatterjee published in Lancet. Let's also zoom this one. There are stories over here. Hemendranath Chatterjee published in the Lancet the results of ORS to treat people with mild cholera. Cholera used to be such a nightmare because the cure was very, very hard. People were like dehydrating and they were dying, and it was like highly contagious. According to this paper, he treated 196 patients with his oral sodium electrolyte solution, rehydrated all his patients with mild to moderate severe cholera. He gave the solution orally, rectally, okay, along with coleus extract, antihistamines, antiemetics without controls, a formula for fluid replacement. Okay, the story is not that interesting. He did not publish any balanced data. He's actually, okay, this is kind of partly summarized here. He did not publish any balanced data. His exclusion of patients with severe dehydration did not lead to any conforming study. His report remained anecdotal. So there is this major worldwide health problem, and then an Indian doctor figures out a treatment solution. That is, now you give sodium chloride with glucose, this thing magically works. At least in case of mild moderate cholera cases. That is his publication. Okay, 1950s. And later on, people will come and say, this was mostly anecdotal, right? So the world is still facing lots of problems. For the next cholera, we don't want to be in this situation where you might have actually found the cure. Let's also open this up. Okay, in fact, Hemendranath Chatterjee is an Indian scientist from West Bengal. So one is to wonder, where are all these biomedical engineers from India, right? He is an Indian scientist from West Bengal, known for earliest publication of the formula oral rehydrated saline. Although his results were published in Lancet is supposed to be the leading medical journal. Okay, they didn't receive much recognition from Western scientists until later. Some argue this was for cultural reasons. Okay, as a treatment protocol included traditional medicine, also because the scientific underpinnings of ORS weren't well understood. So this is a very interesting case where the scientist found the cure. He reported the results. How he reported it, right? And then how much of it is considered scientific? Right? Scientific underpinnings weren't well understood, right? They could always paint this picture if you don't understand it sufficiently enough, right? That is a kind of situation we want to avoid. One of the reasons to deep dive, this is supposed to be a Thursday session, but we are doing on a Friday. One of the reasons to look at this current affairs is because the next Hemendranath Chatterjee shouldn't be put in such a scenario. Okay. So as biomedical engineers, at least some of the most important things like ORS, you should be able to understand very, very well. And now, so that is at least that's the objective of it. I'll give you a much better picture than that in the summary. Okay. And towards that, we also need to understand one more scientist here. Okay. Okay. So Crane, Robert Crane. Okay. He gave a mechanistic explanation for why salt and sugar work together. So essentially, notice you have the digestive surface. Okay, so there is this. This is the gut lining. These cells typically go by the name enterocytes. Okay, so the enterocytes is one example where the cell on the intestinal lumen, the functionality is different. On the back side of the cell, the functionality is different. Same cell, but depending on the sign, it functions differently. Okay, so these are known as enterocytes. Roughly, he has shown that here. What is saying is that this glucose and sugar kind of enter together. Okay, they don't enter things separately. And interestingly, notice he has shown this in a piece of tissue paper. Probably he was preparing for a talk or something like that, and the mechanism he tried to get it in a piece of actually a napkin over here. So any explanation that fits a napkin like this is actually, certainly beauty to marvel. Okay, that is one thing. So again, Robert Crane should be given some credit because for describing glucose sodium co-transport mechanism. Okay, so I'll kind of explain this, and once we understand glucose sodium glucose co-transport, be able to say what are all the sodium potassium chloride, what are their roles in the body. They have very specific individual roles. We want to be able to describe that, and to some extent, why should the composition of ORS be this? Okay, there are three compositions given: standard, low osmolar, and then acceptable. Why should it be in this ratio? Okay, that we want to give a reasonable explanation. And time permitting, there are many other things that we want to deep dive into. Okay, so at this stage, notice understand this fluids inside the body. Okay, have roughly 67% inside the cells. Okay, rest of them is outside of the cells. Outside of the cell means couple of places in the human body. This could be interstitial fluids. This is spaces in between the cells. Then you have intravascular fluid. This is blood plasma as well as lymphatic lymph. Okay, so both are vascular systems. So you have intravascular fluid, that is blood and lymph. You also have CSF fluid, cerebrospinal fluid, that is but is a small extent. So most of the fluid water in the body is inside the cells, 63%, 67%. Rest of them outside of the cells. Outside of the cells also, there is a very clear structure. Okay, that is one thing. And now, if you look at this, of gate questions show up from this picture. Okay, we might have seen this in anatomy physiology, but let's look at this again. You have the cell membrane, that is a phospholipid bilayer. Notice you have a round shape, right? This is typically the hydrophilic end. This end likes water. The tail doesn't like water, hydrophobic. So there are two layers sandwiched like that. That's a cell membrane. And on either side of the cell membrane, you have, the right hand side here in this figure, where there appears to be more potassium, you have potassium is K plus ions, right? You have the intracellular region. The side in the the left hand side in this figure, you have more sodium, Na plus ions, lots of chlorine ions. This is the extracellular space. Okay, space outside of the cells. This is typically the interstitial space. Okay, one thing we can quickly notice here is right hand side inside the cell, left hand side outside of the cell. Outside of the cell actually looks like the ocean, right? Evolutionarily, the cell was trying to differentiate it from the environment, which is the seawater. It serves as an analogy that is more salt outside, which side extracellular space you have more salt, NaCl, sodium chloride outside. It works as an analogy, but evolutionarily, this is how the cell evolved in water, right? In oceans. So you can also see that here and still retained, okay, to this state. So there is typically more sodium outside, more chlorine outside, more K plus outside. At this stage, okay, more salt outside, let's say due to evolution. But what is potassium doing here? What is chlorine doing here? We want to kind of reasonably explain that, okay? Now, let's also look at this figure. Yeah, notice here in the left hand side you have the small intestine. This is where the food and all that is there in the left hand side here. Okay. Now these cells are called enterocytes. This is a lining of the intestines. Okay, separates the intestinal lumen where fluid gets digested to the body which is there in the right hand side here, labeled as capillaries in the right hand side top. Okay, and the whole idea is only sodium and glucose can enter this layer together, not alone. Like a movie, like a date night movie, sodium and glucose is like a pair. If they go watch the movie, they go together, else they don't enter. Okay, after entering, a couple of interesting things can happen, but at least in the movie theater they enter together. Okay, something like there is something called a sodium glucose co-transporter, SGLT1 here. This movie ticket, in fact, let's say the names in the tickets are kind of specific, so only people with those names can enter, and only it's a couple movie. In fact, many bars and all that have this rule. Only couples can enter, right? Singles cannot enter, and couples, their names, you have to be a sodium and glucose, only you could enter. So this SGLT is like that kind of an entrance. Okay, so which is why you need this combination. But to understand the mechanism, we'll have to take a step back. Okay, so let me share a different file. So I want to start with one question. Okay, hey, one second. I had a napkin where I had notes. 1 second. That's it. That kind of flied away. Yeah, this is, I don't know if you can see this is my napkin for the class today. So the first question that I want to get started out with is this. Before we even understand the sodium glucose co-transport and all that, I have a question for you. Does anyone know how does a lift work? Lift mechanism. While I wait for an answer, let me give you two scenarios. Case one, and case 2. Two scenarios. Okay, you have a pulley and the pulley, this is connected to a hand crank. Using this crank, you can essentially rotate this and rotate the pulley. You can rotate it. So that's a hand crank. That is just a crank handle. In the first case, you have a rope. Say this is kind of wound around the pulley. Okay, and a weight is there. Second case, also you have a rope. This is wound around the pulley, but the other end of the rope, the first case it's tied to the pulley itself, but here this is loose, and you have another weight here. Again, you have a handle crank to rotate this. So now I have two questions. If someone asks you to go lift the weight, red color weight, which case would you prefer? Question number 1, which case would you prefer? 1 or case 2? Question number 2, what kind of a mechanism do you have lift, the building lifts have? So Shraddha says, 1, sir, I want to reduce, I want to do exercise, so I'll prefer case 1. So now the objective could be different for different people. One case you want to increase, you want to work out, right? Case 1 is good for working out. Case 2 is more efficient. See, notice in any movie scene and all that, imagine these are, this is, let's say this is the villain and this is the hero. Okay, the hero can easily fall down if it's tight like this. It's very hard to lift this weight upward like this. The thing is, part of the effort you need to rotate this comes from this weight, both are acting, both weights, gravity is acting downwards, but this force is aiding the system, aiding the red weight move up, aiding the blue handle easily crank. Okay, here there is no aiding mechanism. It's good for workout, but it's not efficient. So this case is actually very efficient. That is, if you put a motor or if you have to rotate this crank, you need to spend lesser energy because part of the potential energy comes from downward movement of the green weight. You need potential energy to move this red weight upwards. Part of that comes from downward movement of the green weight. So this is efficient. And this goes by the name counterweight. A lift makes use of this mechanism, counterweight. Lifts are kind of efficient, okay? Counterweight. This is a very simple system, notice. Now, let's see again, if that is slightly, the crank and all that is slightly difficult to understand, let me give you case 3, case 4. Okay. Now, yeah, let me again, this idea is to generalize to this to other systems. Let's think of a seesaw. Okay, so now in this case there is one person playing seesaw, and then in the second case you have two people playing seesaw. Which cases which case is works and which case is playful, right? This is, of course, this doesn't make sense, but we do have people in parks who try this, right? So notice, case 4 is like case 2, and case 1 is like case 3. Analogy works reasonably well because the energy is involved, potential energy is involved, are fairly simple. In fact, if this person is sitting here, he wants to go up, he has to do all the work. This person is sitting, he has to go up, he has to do all the work. Okay, his legs has to do all he can go up momentarily, but he has to do all the work. But in this case, okay, the work is kind of split between the two parties. So this pairing is very efficient, right? The CISA case should kind of make it reasonably self-evident, right? So again, you can clearly see that somehow if you pair it, it's very efficient. Now, there is something in the cells that play the role of this green counterweight. In fact, the green is a counterweight here. Let me write it in the letter green. Okay, something in the cells that plays the role of the counterweight. But here change is movement up and down against gravity. Change could also be movement up and down against concentration gradients. There is a potential gradient, gravitational potential gradient involved here. There could also be a concentration potential gradient. You can try to go upstream, downstream, and all that against the concentration flux. Things usually move from higher to lower concentration. You take salt, you put it in a water, it kind of dissolves evenly throughout, right? It moves from higher to lower. So there is also a counterweight in the systems. So Na plus is a counterweight. This is nature's design to make sure your transport in and out of cells is efficient. This is the role of sodium, primary role of sodium. Okay, most counterweight for transport, but we have to be careful. This is again, if you have to be slightly careful, this is movement in and out of cells. Okay, changed. Electrochemical forces. Typically, there are chemical gradients, there are electric potential gradients. So electrochemical gradients. Idea is the bottom line is slightly difficult to make sense of. That is why I'm giving the analogies. Hey, this plays the role of counterweight. Counterweight somehow, if things are tied to sodium, the transport gets efficient. So in case 4 is more playful, more efficient. Somehow the green person is sitting in the other side, right? So transport gets more efficient. Say transport of not just glucose, transport of most ions and molecules are typically tied to sodium. That is the primary role of sodium. Okay. When it's tied to sodium, that is, as a pair, things are let in, let out, things are made efficient. Okay. I've given two analogies for the efficient cases. So that's the role of sodium. So there are different doors in the cell membrane. One of them is this sodium glucose door. Then there is a sodium, anything you name, typically there is a combined door. Okay, sodium, potassium, for example, Na plus glucose, Na plus K plus, right? So Na plus K plus, the list goes on and on and on. So you could roughly say sodium is a co-transporter. Its primary purpose is a co-transporter. This is why cells spend a lot of energy, a lot of ATP to put lots of sodium outside, because sodium is trying to get in. Let me also show that in a picture. Salt is a co-transporter. In fact, salt is a transporter. Okay, that's a role. That's a role of sodium. Okay, co-transporter. Notice if you take the cell membrane. Okay, you have the hydrophobic, hydrophilic ends. All right, this is extracellular, and then you have intracellular. You have more sodium outside. Here you have Na plus is very low inside. There are specialized channels here that actually put sodium outside. That use energy to send things outside. Expanded, put Na plus outside. So now this sodium, which is very, very high, is dying to get in, because inside it's very low. And when it gets in, something along can, tag along with it. Like glucose can easily tag along with it if it tries to get in. So by default, one is there is an evolutionary reason why we have more salt outside, more sodium chloride outside. There is also a functional reason why we have more sodium outside, because you have like ready gradients established. The sodium is trying to get in the normal. So for this you need energy. For this way movement you need energy. Other way movement, sodium is waiting. Let me go that way, right? Just waiting for an opportunity. The opportunity is a gate opened by a co-transport molecule. It opens up, only sodium goes in. Sodium comes here along with the the glucose molecule or something like that. Sodium is waiting for that, waiting to get in, and then the cell is spending energy to put it outside. This is point number one. Okay, so sodium is a co-transporter. Its primary function is to make transport into the cell specifically more efficient. Okay, because out of the cell, transport into the cell, at least for now, let's say into the cell more efficient. Point number one. Now the question is, what happens during cholera? Again, if you look at this gut lining, this is intra, extra. There are some special gates that control movement of urine. And these gates have two states. There is an off state, there is an on state. In the on state, chlorine will be sent out. In the off state, chlorine will not be sent out. Okay. In the on state, chlorine will be sent out. Sent out the on state. One of the toxins, Vibrio cholerae bacteria that causes cholera, right? The toxin turns on the switch. Okay. So the cholera is essentially turning on the chlorine switch. The sequence is something like this. Cholera toxin turns on switch, it turns on chlorine minus switch. Okay. Now, what would happen because of that? Chlorine leaks. Leaks out into the gut. Where is it leaking out? Into the gut. Now the cell is losing a lot of negative ions. Right? If positive ions don't leave, you are going to have issues soon. Okay. So the next sequence, so this is actually almost sequential. What this leads to is positive ions follow suit. The positive ions we are talking about Na plus, mostly, okay, and K plus follow suit. Suit and N, like they follow chlorine because else charge neutrality is lost. Into the gut. If they go into the gut, they are going to be sent out in the stools. Okay, watery, rice watery diarrhea. Now, so this is why was this happening? This is because of charge neutrality. A lot of chlorine is getting released. Unless positive ions move out, there is no neutrality. Now, because these ions moved, now all of a sudden, the amount of ions inside the cell is very little. What happens is, the third is a disaster. Water follows suit. There is something called osmotic balance. Concentration of solute on either side should be balanced. Charge should be balanced. Concentration should be balanced. Okay, something called osmotic balance. H2O leaks into the gut. So this is what happens in that cholera. This is roughly the schematic of the effect of cholera toxin on the body. It turns on chlorine switches, the switches that control movement of chlorine. It puts them on hyperactive on mode. Chlorine leaks. The chlorine is negative ion. To make up for it, positive ions will leak, sodium and potassium. Okay, and then water also leaks. So this is what we refer to as dehydration. Loss of osmotic and ion balance. It's not just little water. It's it's water and ion imbalance. That is a better way to put it. Okay, water, and often this is mistaken, ion imbalance. It's not just water imbalance. Now, fortunately, this process, the cholera toxin only affects this sodium glucose transporter. I showed you this SGLT1, sodium glucose co-transporter, right? That that channel is still intact. Across the cell, there is a particular channel by the name SGLT1. This is a sodium glucose. Sodium and glucose can only enter through this door, Na plus glucose channel, still intact. Still intact. That is, if both sodium and glucose are there in the gut side, this is the gut side, this is the capillary side. Okay, if there is both Na plus and glucose, it will be sent in, because sodium is, the point of those channels is to send sodium and glucose together inside. So what happens is, if you just give sodium, this channel will not let in the pair, movie pair. Okay, or a movie is fine. So sodium alone goes to the movie. No, you have to come with glucose. Go get your pair. Glucose alone, show where is your pair? You need to go come with your pair. Bring your sodium. So which is why now if you give both sodium and glucose together, sodium can enter. ORS solution. Okay, this has Na plus, K plus, chlorine. This also has some bicarbonate. Okay, this is where we gave the citrate, sodium, potassium, chlorine, calcium, magnesium. Yeah, at least you pair glucose. I was wondering. So the overall solution has this. So what happens is, the moment you give that, Na plus and glucose enter. Okay, now enter inside, that is cross the enterocytes. Okay, let me also put it what I mean by that. Cross enterocytes, the cell lining in the gut, enterocyte. Okay, now once sodium and glucose enter, now you have solute concentration higher inside, that is, this is in the capillary side, higher in capillary side. Again, enter, this is cross enterocyte, goes to the capillary side. Let me be from the gut, inside the gut lumen, goes to the capillary side. So solute concentration is higher. Now so this is what we refer to as osmotic imbalance. The concentration should be the same inside and outside. Else there is a gradient and there will be movement. So now, because concentration inside is higher, because more sodium entered, H2O follows suit. H2O can easily come in and out. Okay, H2O is like now this. These are like ghosts. It doesn't bother about channels, this, that, and all that. Water can easily move in and out. Okay, water everywhere. Interstitial, extrastitial, interstitial. So this doesn't need any special tickets or anything like that. Water is a VIP as far as the living system is concerned. Also enters in enters in again. I typically this is crosses the it gets into from the gut, it gets into the capillaries. So sodium entered because of glucose. Sodium concentration very high in the body now, in the cells or in the capillaries interstitial fluid. Okay. Now water from the gut also enters to make up for it. And the moment water and sodium enters, okay, now plus also at this stage balances. At this stage typically balance things are balanced, rehydrated. And in this case, what I need, what I didn't mention as positive ions. Typically, there's a sodium leaking out, but this is not a lot that is we are losing. Anyways, these are not in the interstitial side. There is only more sodium in the interstitial space. Potassium is inside the cells, right? So only sodium leaks into the gut from the interstitial to the gut. Okay, potassium is deep inside the cell membrane, safe, so it doesn't leak that much. But just in case, that also is like kind of lost. Okay, K plus balance is also established. And the body, once the problem is dehydration. And the bacterium itself is typically the body fights itself, the immune system. In the process, this osmotic balance, ion balance in the body is lost. If you support that using ORS solution, the body recovers, fights the infection. So in very rare cases, the immune system is very weak, or the bacterium is very strong. This antibiotics are given, but typically the body fights the infection. You just need to support it. Once you support it, the body recovers. And now children can now get back their life, right? So the doctor is happy, the children is happy, parents, everyone is happy. So this is the sequence that happens from the toxin, chlorine leakage, followed by sodium leakage, followed by water leakage. You give sodium and glucose together, sodium enters, water follows suit, you get rehydration, followed by recovery. Okay, now this is one thing. And the important lesson here is that sodium is a co-transporter. In fact, sodium is, I would like to say, is the co-transporter. Makes transport in and out of cells effective. And to hijack, or to hitchhike into one of the available vehicles, sodium glucose vehicles, give sodium along with glucose. That vehicle is available, that will take care of, that will transport itself. So this is how ORS solution works. And notice, if you look at this, so sodium and transport, some numbers here. Yeah, so this channel is called Na plus K plus sodium potassium ATPase. Okay, this is in the ratio of 3:2. This was actually a gate question. There is more sodium outside, more chlorine outside, more potassium inside. That was also a gate question. Okay, what is the ratio in which sodium potassium will be actively transported? So this is 3 sodium ions to 2 potassium ions. So that That was a question once. Okay. And this is in the ratio 1:1. Glucose, 1 sodium. So which is why your ORS solution will have similar moles of sodium and glucose. Lefty, 75 millimoles per liter, 75 millimoles per liter. Potassium is not lost to the extent in which sodium is lost, but still it's lost, so you kind of replenish it. At least 1/4 of sodium, potassium is given. Still, it is kind of lost, it is given. And so another thing I didn't get to talk about today is this acid balance, acidosis that is going on. This keeps, blood is slightly alkaline. The primary reason is bicarbonate. If there isn't bicarbonate, the blood gets acidic, acidosis. So there is also an issue with that, but today I didn't discuss that. But my question right now was, sodium is a co-transporter. It makes transport efficient. Yeah, analogy, mechanical analogy is that's a chemical movement. Now, is a mechanical analogy. Does that analogy make sense? Quick question. And before I, say other things that I said, okay, now we'll do. That is, hey, what is the role of different ions from this perspective? Let me do a quick poll. ORS mechanism and Na plus analogy sense. Okay, let's just make it simple. No, I try to make it reasonably. This is not too complicated. Okay, so now the majority says so that's actually two stated. So ORS mechanism, what happens in this cholera, and then this co-transport analogy. Okay, now so that was reasonably clear. Now let me shift the gears and view like what the other ions are doing in the sun. Okay, so notice quickly. Let's look at this. Yeah, if you have any questions, put it in the chat. Yeah, low osmolar typically notice sodium glucose same number of moles per liter, millimoles per 75. Potassium is lesser, and then you cannot give sodium and potassium alone. You need something something to bond those ions, right? That's why this is a solution of salt, sodium chloride and potassium chloride. And citrate gives you bicarbonate. Okay, citrate gives you bicarbonate. If you look at the carbonate, I showed screen. Let me share a screen. Carbonate is this COO group. Okay, COO bicarbonate ion shown over here. Okay, and H2CO3 in that. So this is carbonic acid. If you remove the proton from that, if you remove H plus from that, it's going to be a bicarbonate. Carbonate. Okay, and citrate will have couple of these groups. Same group will be repeated. That acts like a buffer. Notice there is a bicarbonate CO group here, CO group here, CO group here. So this acts like a buffer. That's why you give citrate, citric acid. Now the question is glucose lost during dehydration? No, glucose is not lost, but if you eat, it won't enter. That is, if you eat glucose water or anything like that, the body will not get the benefits. Glucose is not lost. Glucose is getting consumed. Energy is getting consumed. The body is in a repair fighting mode. It gets consumed. That is one thing. It doesn't leak into the gut. But the problem is, if you just give glucose, it doesn't enter. So you need salt. Okay. Even glucose is lost during dehydration. Glucose is metabolic. This hydration is ionic. Ion water balance. Okay, what is the concentration of the solute and the solvent? Right? Dehydration pertains that question. How much of energy do I have and how much of energy am I consuming? Glucose has to do with that energy consumption balance. This has to do with concentration balance. So those two are very different. They are coupled here because of that co-transport. Sodium enter, water will enter, but for sodium to enter, only one available vehicle takes glucose along with sodium. Okay. Okay. Now let's get back and give me this last bit. Okay. Okay. Now you looked at a couple of. This is important also from a gate perspective. There are also numericals from this. This we will look at. This is also a good time to look at it because this is also this is where the origin of biopotentials. Where is this membrane potential coming from? Because of this ionic, there is more sodium outside is trying to come in. So why is it not coming in? Something else is actually stopping it from coming in, right? So some forces match up. That is where this biopotentials, resting potential, and all of that show up. There can also be numericals based on that. Nernst potential. Okay, we'll look at it eventually, Goldman equation and things like that. But for now, let me fill this so that you appreciate some of these ions better. Okay, so point number one, Na plus, there is more salt outside of the cells, fine. Na plus, co-transport. So that is, it makes transport efficient. Transport efficient is the role of Na plus. So what is K plus doing? So again, analogy wise, so if sodium is going in and out to transport things, concentration differences in and out. Now this osmolarity, this is known as osmolarity. Osmosis, osmolarity. Okay, let me write that word. Amount of concentration of solute and solvent should be balanced on either side. If sodium is moving in and out to transport things, this osmolarity balance is affected. Potassium's role is to re-establish this solute solvent concentration differences. Total number of ions in the water, the solvent is water, total number of ions. The sodium has changed, if potassium makes up for it, in the total number of ions still remain balanced. Okay, so K plus maintains this maintains osmotic balance. While sodium is doing its work, potassium is aiding sodium do the work. This is why this Na K plus ATP the active channel is so important. So much ATP energy is spent moving potassium in and out in the ratio 3:2, right? So maintains osmotic balance as while Na plus is doing its work. Osmotic balance. While Na plus is moving in and out. This is why these two are deeply coupled. So sodium is a co-transporter. K plus maintains osmotic balance. You could actually think of K plus as a counterweight for the counterweight. Sodium is a counterweight. I would like to call it counterweights. Counterweight is like a dream within a dream, right? Question within a question, answer within an answer. We'll put counterweight within a counterweight. Sodium is already a counterweight. There is a counterweight for sodium, that is your potassium. Or in simple terms, it maintains osmotic balance. Its role is to maintain osmotic balance while sodium is like now doing its work. What is chlorine doing? Chlorine, these two positive ions now can easily change tip charge neutrality. You don't want, we are not trying to make, this Spider-Man movie villain, Electric Man. I don't know, Electro Man or something like that, right? So eels do have this capability, but in general, our cells, we don't want, if you're touching your friend, he shouldn't get, you don't want him to get electrocuted, right? So there is charge neutrality, both in and out, unless there is specially designed for that, okay? Typically, most places in the body, the charges in and out, it's neutral, charge neutral, zero charge, zero net charge, okay? So our hairs are not normally rising and all that. So our body is charge neutral. This is balancing Na plus and K plus. Charge neutrality. Neutrality. That is, while sodium and potassium are moving in and out, something should balance the charge. Charges shouldn't go berserk. Okay. While Na plus and K plus are moving in and out, Cl minus, again it moves in and out, maintains, in fact, Cl minus movement maintains charge neutrality. So potassium is for osmotic balance. This is for charge balance. Okay, maintains charge neutrality. Charges are neutral doesn't mean the electric potential is the same. Electric potential is something very different. It can be potential voltage could be negative inside. It's a very we are not talking about the Coulomb and voltage are two different things. Okay, potential could be negative inside, but net charge is neutral. Okay, that is one more point. And then so this is why typically when you model, you at least consider sodium, potassium, chlorine to consider a cell's balance. Okay, and if your body is running out of water, if you just drink water, your kidneys typically gets overworked. Your body is going to lose more water, because to establish ionic balance, you need water and all these ions. Okay, and typically if some of these ions should enter, at least in case of abnormal cases, you need some little glucose along with it. Okay, that is one thing. And let me also conclude this with two other important things. So bicarbonate, we'll have to have a separate discussion, but for today, so there are some interesting questions that I want to, so in 5 minutes we'll wind up. Calcium 2 plus. So why does calcium get deposited in atherosclerotic plague, in blood vessel walls and all that? Why does it get deposited? Have any of you thought about it? Why does it deposit in walls, aortic wall? There is calcium deposition. Why does that happen? What is the role of calcium? There is too much calcium imbalance. What will happen? Someone will get into a tetanus and all that. Their muscles will be like active, will be like stuck and things like that. Okay, so calcium, in very simple words, it's a cell signal molecule. Example, it tells you whether a muscle should start contracting or not. If there is calcium, muscle will start contracting. Cells will signal, hey, come on, start your contraction by controlling concentration of calcium. The moment there is calcium, the muscle cell, myocyte, will contract. Heart muscle cell, any of that. Again, calcium also plays a role in it. This is very important. It's a signal. This is like a light. Yeah, actin myosin cross bridge needs a molecule to function. That is correct. By that we mean, so Ca plus acts as a signal for the bridge to form, cross bridge cycling. Yeah, that's exactly correct. Okay, so this is a cell signaler. Should the muscles contract or not, it is being signaled by calcium. That is one role. Neurotransmitter, this synapses, how signal transmits from one neuron to another neuron, that cell signaling is also controlled by calcium. Okay, calcium and calcium pumps. Okay, that is a important. It is an important cell signal molecule. And then we have magnesium. Magnesium 2 plus, or magnesium, I suppose 2 plus. Okay, magnesium plus, actually periodic table is in a different desk. What is magnesium? What is its role? Magnesium's role in the body, unique role. Magnesium 2 plus. What is the role of magnesium 2 plus? With this we'll finish. This actually controls enzymatic activity. Okay, this magnesium is a cofactor in many enzymatic reactions, which means most of the reactions in the body. Okay, so this is metabolic. Many metabolic reactions can happen only in the presence of magnesium. Metabolic cofactor. Cofactor, or it's not a coenzyme, and it important function is it does mitochondrial. It helps the mitochondria do its job. Not mito. There is a theory that mitro, based on the DNA of mitochondria and all that. So mitochondria is likely a separate life form. Then nature found a symbiotic relationship between our cells and mitochondria, so that mitochondria can live in our cells. Okay, get supply of food, and then in turn mitochondria can give the cell supply of energy. So there is a symbiotic relationship between a cell and a mitochondria, which likely existed as a separate bacteria entity. Okay, so there is a difference. So this is a mitochondrial helper. Yeah, so most things. So this is a cofactor. This by the cofactor, I mean this is a helps many enzymes. Okay, and then for few other things, zinc you would need for immune function. The list goes on and on, but I think the important one here, I think where we should stop here is carbonate. Because this balance one more. So pH balance. There is osmotic balance, there is charge balance, and then there has to be a pH balance. Blood, for example, should be slightly alkaline. So what is the role of that? That again we'll see if I can get you. Okay. So, but at least right now you're starting to see this. The moment they have specific unique roles, you start appreciating them better. And of course, if there is a deficiency, if you eat too much salt in your food, your kidney is damaged. It is the thing that is trying to maintain osmotic balance in your body. Your kidney is gone. Easy way to damage your kidney: take too much salt. Easy way to actually improve your heart function: reduce lower salt intake, because now your kidney is functioning well. If your kidney is functioning well, your heart is functioning well. So you can, there are many things that we can discuss here. And if you take too much potassium, your heart will be affected. Okay, because now your heart's electrical activity depends on potassium, and that's very sensitive to potassium and things like that. So now you can go, if you know the specific roles, you can actually understand physiology and pathology better. Okay, fine. That is the objective. I already got a minor feedback on whether the objective of the meeting is met. If there aren't any other questions, we are slightly over time. Okay, I'll try to find another topic where that is interesting, and where we also get to learn. Many of these things are there in the GATE syllabus. Some of the concepts, some of the questions are recurring with respect to some of the things we discussed, right? I mentioned some GATE questions. So these are also important from my GATE perspective, but genuine curiosity and motivation and all that shows up better if you approach it this way. Okay, that is what I try to do. Fine. So this is done. We look for another important, interesting topic next time. Okay. Until then, I will see you again. Have a good one. Take care. Bye, everyone. Happy Onam. Everything. Okay.